Hello, and welcome to the PhycoCosm tutorial. My name is Alan Kuo, and I am a data scientist on the fungal and algal genomics team at the Joint Genome Institute, or JGI. In this series of video tutorials, our algal team will discuss the various omics data and analysis tools available on our algal web platform, PhycoCosm. In this first part of this three-part series, I will present an overview of our various tools for navigating to the data available in PhycoCosm. Let's get started. At the JGI, we perform genome sequencing, assembly, annotation, and analysis at scale to support our diverse user communities, including our collaborators who supply DNA and RNA samples for sequencing. My own team at JGI specializes on the annotation part. We perform both structural and functional annotation using an automated annotation pipeline for high throughput processing of algal genomes. There are many ways to predict genes, including ab initio modeling of exon-intron structures and modeling from RNA alignments and from protein alignments. The JGI annotation pipeline uses all three methods, resulting in multiple gene models per locus. Each model is next annotated for function by searching GenBank for orthologs, or searching PFAM for domains, and by other methods. These structural and functional characterizations are used by the pipeline to select the best model at each locus, to be added to a filtered model set. The filtered model set is used to seed a gene catalog, which is released as an initial gene complement for that genome. Each genome data set we generate is pre-released as a genome web portal for a limited time to expert collaborators from the research community for assessment and improvement. And then the data set is released to the public for the benefit of the entire research community, ready to be used for the comparative genomics and multi-omics analyses for which PhycoCosm has been created. The great advantage of PhycoCosm is its gathering of so many genome portals in one place. Many of these genomes were sequenced and annotated by JGI, but many were not. The comparative power of PhycoCosm is enhanced by the incorporation of each additional genome. And in pursuit of such enhancement, we actively import data sets published by the algal genomics community. In the past, these were imported as is. But since these come from myriad sources that take a variety of approaches to annotation, we are beginning to apply our pipeline's filtering capability to these external gene model sets to make them more comparable to JGI gene catalog sets, as well as more comparable to each other. With or without filtering, we always preserve all the original external annotations in our portals. Now let's stop these slides and look at PhycoCosm itself. Let's direct our web browser to phycocosm.jgi.doe.gov. At PhycoCosm, we have developed a variety of ways to connect to the data you're interested in exploring further. On the left sidebar of the homepage, you will also see announcements about recent genome releases, details on upcoming conferences, and information about JGI Community Science Proposals, or CSPs. A CSP is the primary way that you, the individual algal researcher, can get your favorite alga's genome sequenced at JGI. If you're interested in submitting a CSP, you can see more details by clicking on this link. The ultimate result of the process to begin with a CSP will be, among other data, your favorite alga's genome. Here we see our most recent genome releases at the time of this recording. Tribonema minus is the world's first xanthophyte genome to be sequenced by Chrissy Mahan and her colleagues at Sandia National Labs. The next most recent portal release is of the genome of the oomycete Hyaloperonospora, a non-photosynthetic relative of heterocon algae that we did not sequence, but we did import. The PhycoCosm homepage is dominated by the cartoon evolutionary tree in the center, the PhycoCosm Navigator. We also have a very useful search function immediately above it, which can help connect you to the organism in which you are interested in. We could type something in here to search directly, but let's instead click on the search button to see more details. When you click the search button, you are by default brought to a list of all the genomes in PhycoCosm. You can pare down this list using the search functionality at the top. You will see there are multiple options which allow us to better trim down this list. You can request to only look at genomes produced at the JGI or vice versa. You can request to only look at genomes that are public or published. The difference between public and published and what it means for data use permissions will be discussed in our other video tutorials. If you already know your alga or algal taxon of interest, the most useful feature of this page is the keyword search. Here you can search directly for the species or taxon you are interested in. Say, for example, you're interested in the class of pinnate diatoms, the Basilaria Phyceae. We now see 5 different species of Basilaria Phyceae, including the model diatom Phaeodactylum tricornutum. 4 of these genomes are published, shown in green with associated references, and 1 more genome is public. You can also search by NCBI taxonomy. Say you want to identify some close relatives to the pinnate diatoms for comparative genome analysis. We can search for all genome data available for any member of the diatom phylum Basilariophyta. Of which Basilaria is a class. You can see that we have now expanded our set to include 9 different taxa, including the centric diatoms. This taxonomy search functionality is based on NCBI taxonomy. So if you're so inclined, you can even use NCBI taxonomy IDs to filter the data. For example, let's zoom out one more step to the kingdom Heteroconta, which has the NCBI taxonomy ID 3, 36, 34. Search for this in our portal and you get a set of 33 protists. Another unique feature of this tool is the ability to search by publication. Our published genomic data are linked to papers using Pubmed IDs. So if you read an interesting paper that published some new JGI omics data, you can enter that Pubmed ID in our system to retrieve all the genomes included in that study. For example, we can try to look for the Phaeodactylum genome paper, which has Pubmed ID 18, 92, 33, 93. The paper also involved improvement of the genome of Thalassioceras sudinata, so that diatom also appears on our list. And finally, if you are fond of a particular author's work, you can search by their name and see all published genomes they are associated with. The search functionality is a great tool if you have a priori knowledge of where to look. However, if you don't have a particular organism in mind and want to visually explore the available data, we have some browsing options for you. The first of which is the PhycoCosm Navigator on our homepage. You can always get back to the Navigator by clicking the PhycoCosm button at the top left of the screen. The Navigator is a cartoon showing a hierarchical taxonomic organization of genomes across algae and their closest relatives. For example, Archaeplastida consists of Rhodophyta, Glaucophyta, and Viridiplantae, which consists of Chlorophyta and Streptophyta, which includes the Embryophyta. Note that Embryophyta are plants, not algae. Though they're not algae, we host a select set of plant genomes because such genomes are invaluable for certain evolutionarily informed studies of algae. For the same reason, PhycoCosm also hosts a number of non-photosynthetic protist clades, those that are the closest evolutionary kin of true algal clades. These are colored gray, for example, the Eomycetes. Each branch node is named and clickable, as is each leaf. Clicking on any of these nodes or leaves, say the Viridiplantae node, will reveal all the genomes we have available at that taxonomic level and below, as well as access to some comparative tools for analysis across this group. One of these comparative tools is called Tree, which provides yet another way to visualize the data in PhycoCosm we will talk about later. At the top left of the Navigator, we have a pull-down menu. This menu is for accessing various groups of genomes. This pull-down menu is color-coded to indicate the kind of genome group. The majority of groups have no color. These are what we call phylogroups, meaning the group is defined by phylogeny. Every node and leaf in the Navigator cartoon is listed here in this group, a menu, as a phylogroup. For example, the Basilariophyta. We also have what we call eco-groups, which are manually curated groups of organisms that share a common feature independent of phylogeny, usually having to do with common phenotypic or ecological characters. These eco-groups are highlighted in blue-green. For example, we have eco-groups. We have an eco-group for Arctic algae, one for seaweeds, and one for various lichens. The lichen eco-groups are notable because they also contain fungi hosted on PhycoCosm's sibling website devoted to fungi called PhycoCosm. Note that algae itself is an eco-group, as algae are a non-phylogenetic set of eukaryotes with a common attribute of photosynthesis, excluding plants, of course. If you're interested in a particular eco-group and do not see an organism you would expect to see there, please contact us and we will see to adding it. To explore a group, you can select it from this pull-down menu, or you can click on that node in the Navigator. For example, let's take a look at the group and node Chlorophyta. The Chlorophyta are the green algae. When we access a group, we land on that group's info page. At the top is a toolbar with various options you can explore. Our other PhycoCosm video tutorials will discuss the various tabs for various levels of analyses, but now let's highlight another way in which you can view the data, the interactive tree. The tree tool provides a searchable and interactive phylogeny for exploring the evolutionary relationships between the members of the group. It is a maximum likelihood tree calculated from whole genome protein sets in PhycoCosm. The tree consists of nodes and leaves connected by branches whose lengths show evolutionary divergence. Each node is the bifurcation of a branch. For example, the classic Evo-Devo sequence Volvox and Gonium are more closely related to each other than to any other organism at PhycoCosm. The color of a node indicates support for that bifurcation. The green color indicates that the support for the Gonium Volvox node is as good as any. Many nodes correspond to taxonomically well-defined phylogroups and are labeled as such. Mousing over a node, say Chlorophyceae, instantiates a green box delineating the clade. Finally, each leaf is a sequenced genome and genome portal, either internal or external. The Chlorophyta phylogroup consists of many genomes. To find one organism, enter its name in the text box and click Filter. For example, let's look for the ubiquitous sea lettuce, ulva. Note that the ulva genome is now highlighted, and many other nodes are collapsed for easier viewing of this target of this genome. Click on Clear to return those other nodes and leaves to visibility. You can customize your view of the tree by expanding and collapsing individual clades. Click on it by clicking on an individual node. For example, we can expand the Chlorophyceae clade like so. And we could recollapse it by clicking on it again. Clicking on the node also invokes an info box for that node. The info box shows details about Chlorophyceae, such as the node's bootstrap support, its distance from its parent node, its NCBI taxonomy ID, and its degree of agreement with the NCBI taxonomy, which in this case is 100%. There are also links to the corresponding NCBI taxonomy page and to the PhycoCosm Chlorophyceae group portal. Our video tutorial, part 3 on comparative genome tools, will show us group portals in more detail. You can zoom from your tree to a subtree by shift-clicking on the node for the desired subtree. So let's shift-click on the order Clemidomonadales to see its subtree. You can zoom back out of a tree by clicking on the up arrow in the info box. For example, we can go back from the order Clemidomonadales up to the class Chlorophyceae, like so. So now we've learned a few ways to navigate a group's phylogeny using the tree tool. Next, let's complete our descent through the Viridiplantae hierarchy, through Chlorophyceae and Clemidomonadales to a single green algal species. You can do this by clicking on a leaf to go to that genome portal, say the model alga Clemidomonas reinhardtii, As a model organism to algae and to protists and to plants, Chlamydomonas has been subjected to genomic, transcriptomic, proteomic, and many, many other kinds of omic analysis. Our video tutorial, part 2, on single genome tools will show us the ways that PhycoCosm can be used to visualize all of that data. This concludes part 1 of the PhycoCosm video tutorial. Today we visited PhycoCosm and learned about the Navigator and the tree tool. You can learn more and in greater depth by watching the next 2 parts of this video series on single genome tools and comparative genomics tools. Thank you for listening.